Okay, thank you very much indeed. Good afternoon, everybody, and welcome to today's live webinar hosted by MSD and AstraZeneca. Today, we're going to be presenting the results from Run 16 of the Variant Classification EQA Summary scheme. Next slide, please. We're joined today, so myself and my co-host, Professor Sandy Deans, from the Director of GenQA, are joined today by I call them a cast of 5 very eminent and well-known scientists within the field of genomics. We've got George Burgle from Manchester, Professor Kathleen Klaas from Ghent in Belgium, Bethan Frugenet from London in the UK, Andrew Morani from Alberta in Canada, and Professor Thomas van Overaan Hansen from Copenhagen in Denmark. And together they're going to be going through and summarising the results from this EQA and providing the evidence to support the decision-making associated with the results. Next slide, please. This introduction obviously is welcome for myself and Sandy Deans, who I'll be introducing in a second. Next slide, please. Before we get going, just a quick outline of the agenda we've got for you today. This is an interactive webinar, so we've got time at the end of it for live Q&As. After I've done the introduction, we'll be doing an overview of the results, just an overview of the entire results from the run, presented by Sandy, and then we're going into the in-depth explanation of the variant classification that we've given to those 5 variants within the context of this EQA. We've got time over at the end for about 20 minutes for a live Q&A, and then we'll summarise and wrap up in about 90 minutes. Next slide, please. Some important housekeeping questions or requests from you, first of all, before we get going. We are recording this, but we are automatically muting your background, the background noise, your microphone. The intention is to make this available for you to re-watch if necessary, or for your colleagues to watch afterwards. Usually takes a couple of weeks for us to go through compliance, but we will make sure that when the webinar is ready for you, we'll notify you through the GenQA and EMQM websites. You can, most importantly though, this is an educational webinar. So we have the Q&A session at the end, as we described already. Please do submit your questions at any time. You can use the Q&A function to do so. And Sandy and myself will be putting those questions to the speakers at the end. Next slide, please. So your feedback also helps improve our performance and our webinar schedule for your interest. We do pride ourselves in trying to make sure that these webinars are highly interactive, but also highly educational for you. So after the webinar, you will receive an email from the AZMSD teams with a link to a short feedback survey. I would encourage you, please do commit the 2 minutes or so of your time to complete it. And help us to improve what we offer and deliver to you. Next slide, please. Finally, our disclaimer. So this is obviously a non-promotional webinar. It has been organised by AZ and MSD for your benefit. The views articulated are those of the speakers and not necessarily those of AZ or MSD. And the speakers have received an honorarium from AZ and MSD to support this webinar. Next slide, please. Right, I'm going to hand over to my co-chair, Professor Sandy Deans from GenQA, who's going to run through the overview of the results, the summary results from the EQA scheme. Sandy, over to you.
Thank you, Simon, and I join Simon in welcoming everyone today to the webinar. So next slide, please. So can you believe it? We're on run 16 of this EQA assessment. So we're going to today focus on the participation in the BRCA1, BRCA2 and other HRR gene variant classification assessment, which was run 16. Next slide, please. So this assessment was run on the GenQA Genie platform. That's our genomic online individual education platform. And we've got many, many modules on there. So I would encourage you to have a look and see if there's anything there that would fit your needs. It's accessible anytime through the GenQA website. It's an individual account and it's available globally all year round. Many of the scenarios are randomly generated, which means that you can, every time you log on and open a module, you're going to get presented with different cases. So you can participate multiple times. And the great thing about it is it's not necessarily like an EQA where you have to wait for the results to be released. On many of the modules, you get real-time assessments, and you can complete this at your own pace. And there's also a training or performance review function for managers and training officers. So have a look if you're interested. So we use this platform to deliver Run 16. Next slide, please. So this is some of the metadata that we collect. The Run 16 was delivered during the month of April this year. We had over 300 individuals registering with 287 participants. And these were from a wide range of countries. So we have 42 different countries represented into the participant list. So we truly are a global scheme, and I think that's the benefit of using such an online platform. So our top countries were Poland and the UK, followed then by Germany, Thailand, Italy, Slovenia, and Spanish participants. And the graph along the bottom just gives you an outline of all the different countries that are represented. Next slide, please. So some of the other data that we can collect through our participation is about which guidelines are being applied, and particularly with the BRCA genes and the HRR genes, there are many different guidelines out there that are available and can be applied. So we see, compared to the last run, which was towards the end of 2025, we can actually see a decrease in those who are applying the ACMG 2015 guidelines only. So you can see a reduction there from 16% down to 13%. And we also see a decrease in those using the ENIGMA guidelines only. Again, that's a slight decrease from Run 15. There still remains to be about 40 to 45% of individuals who don't state which guidelines they use. And I would encourage you in future to please do this, because it will become even more important as the new ACMG version 4 guidelines are published next year. Next slide, please. We can also work out who is actually participating. So we asked about you to self-determine what your specialty is. And I guess no surprise really, about 51% are from either clinical genetics or genetics departments themselves. We have still, again, a small, about quarter of participants who don't actually list what they are or say that they're other. So I think we need to do a bit of a deep dive to work out what type of background those individuals have. We also ask you what your area of expertise is, whether you're a scientist or a medic, etc., pathologist. And again, we can see that the majority, not surprisingly, as we see with previous runs, that the majority are experienced scientists. Although, again, there's a large number, another quarter, who say that it's not listed. So we will take that upon ourselves to try and determine a few more areas of expertise to add to your list. Next slide, please. So these are the 6 variants that were presented for Run 16. As we usually do, we try to vary the expected classification. So we range from benign right through to pathogenic and all different classes in between. We also try and vary the types of variants, so SNVs and small indels, and we also had an 2 exon deletion in the PALB2 gene we included. So here you can see case number 1, variant number 1. The classification can change depending on which guidelines you apply, but they're both within the same clinical management group. So they're either likely pathogenic or pathogenic. Next slide, please. So these are the results from Run 16, very high level, and all individuals will get a copy of the EQA Summary report that will summarise all participants and the applied criteria, as well as your own individual feedback. But here you can see on the table on the right-hand side that our percentage of individuals correctly classifying all 6 variants has dropped. In fact, it's halved compared to Run 15. We don't quite know why that is, and we're going to do more of an in-depth review of which variants were particularly challenging, and that I'm sure our experts will touch upon during the webinar. However, we do see a larger number of participants receiving correct classifications for 5 of the 6 variants and also 4 of the 6 variants. So I think the standard is improving, although there's still some way to go until we are all classifying all variants correctly or as according to the consensus. Next slide, please. So now I've got great pleasure in handing over to the first of our 5 experts on the panel. George is going to run through variant 1 and the in-depth analysis of the data. Thank you, George.
Thank you very much, Sandy. Just checking if you can hear me.
Yes, we can.
Excellent. Thank you very much for confirming. So, yeah, I will cover variant 1 for you today. And as Sandy explained, it's the only variant on this scheme that actually differs slightly between whether you're applying ENIGMA guidance or CanVIG guidance, and we'll go through that, but both still within the same clinical actionability bracket. So the first variant is a missense variant in BRCA1, and it is within the functional BRCT domain. Looking overall on the variant itself, it's absent from NOMAD version 2 and 3 in the non-cancer population, but it is present at a very low frequency in version 4, including a control in the UK Biobank dataset. There have been 10 submissions on ClinVar, achieving an overall 2-star classification of pathogenic, likely pathogenic, and most in silico prediction tools, including Revel and Basedel, predict a deleterious impact of this variant. So following the ENIGMA VSEP guidance, we reached a class 4 likely pathogenic classification, applying PS3 strong, PM2 supporting, PP3 supporting, and PP4 moderate. However, using the CanVIG guidelines, we achieved a class 5 pathogenic classification, applying also PS3, PM2, PP3, similar to the ClinGen ENIGMA guidance, but PS4 strong on top and PP4 supporting instead of moderate, and we'll try and tease out the reason behind these differences between the different guidelines. So I'll run through the overall results, and you can see that the participants yourselves have done extremely well with this variant. We consider both class 4 and class 5 as correct and expected classifications, and 98.3% of the participants got that correct. Only 5 participants did not get likely pathogenic and pathogenic. So that's an excellent overall classification for this variant. So going through the details, I will start with the evidence criteria that both guidelines agreed on, and then we'll focus a little bit more on the ones that they differed slightly in between. So starting from functional studies, as you hopefully all know very well, we use the Wernicke framework, which is the published from the ClinGen SVI group in 2020, on how to interpret or how to assign strength when you're applying functional evidence, structured functional studies using either PS3 or BS3. And for this variant, as you can see from the summary here on the right-hand side, there are multiple studies published that have investigated this variant, all of which are high quality, very well-known ones, including Finlay and Bowman. And these are studies that have been assessed using the Wernicke framework as achieving a strong level of PS3. This summary here is available on CanVIG. I'm sure many of you are familiar with it, and we have presented in previous schemes the database itself. I would recommend watching the recording and using this website, as it provides a lot of valuable information, and you can see a lot of functional studies scoring on there. But anyway, this variant has multiple studies pointing towards loss of function or deleterious impact, and it meets both ENIGMA and CanVIG criteria for a PS3 strong. Most of you have applied that, 87% of participants, and few applied it at only supporting or moderate. Then going to PM2, so that's based on the population databases. It meets the criteria for ClinGen because it's absent from NOMAD version 2 and 3 in non-cancer population, and because it's present at very low frequency, including one case in UK Biobank, it is also meeting PM2 supporting. And again, most of the users, 73%, applied it at PM2 supporting. There have been few moderate, but very few also strong and very strong, which should not be used because it's not an option. And as you all know, PM2 may not be or will not be there in future iterations of guidelines. In terms of in silico predictions, PP3, the variant achieves the same using both ClinGen, ENIGMA, and CanVIG criteria. Both Revel and BaseDel predict a deleterious impact of this variant, and it is within the functional BRCT domain, so it meets the criteria in both guidelines to be used at PP3 supporting. Again, most participants used it at the correct strength, and very few mistakenly applied it at strong or very strong. Again, I wouldn't, this should not be used at a higher level than supporting. Now, when we come to some of the difference between the guidance, I'll try and explain the reasoning, the data that's available, and why it may be applied differently between different sets of guidance. So we'll start with the ENIGMA guidelines, and starting with the guidelines on PS4. So PS4, as you know, is the prevalence of the variant in affected individuals when compared to controls in a case control study with matched ethnicity and country control dataset. And the variant should be statistically significantly more prevalent in cases compared to controls, achieving a odds ratio of greater or equal than 4 with a very tight lower confidence interval that doesn't overlap with 2. Now, importantly, the ENIGMA guidance does state that if a study is presenting odds ratio alongside case control likelihood ratio, then you should use the case control likelihood ratio instead of the odds ratio within the PP4 criteria. Okay, so we'll come to that in a second. So that's an important distinction to keep in mind when you're following the ENIGMA VSEP criteria. And it's also important that the definition for case control study, at least as we understand it and based on communication with the ENIGMA VSEP, it has to be within a specific case control study setting. And that's why some of you are familiar with the National Disease Registry service data from the UK. We don't use NDRS compared to UK Biobank as a case control if you are following ENIGMA VSEP. So the study that we used to look at PS4 and PP4 within ENIGMA mainly was this paper published in 2025, Zanti et al., and it looks at more than 400,000 women with BRCA1 and BRCA2 variants for classification. And if you go to this specific variant in their supplementary material, you can see that it achieves a very high odds ratio of 4.6, achieving all the requirement to meet a PS4 strong criteria. However, they also present the case control LR method that I've mentioned in the previous slide, and that achieves 13.9, which qualifies for a moderate piece of evidence under PP4. And like I explained before, the guidance does state specifically if case control LR is available alongside the odds ratio, that should be used in preference to the odds ratio. And they do specifically refer to the Zanti et al. It was in preparation at the time the guidance was last updated, and they recommend using the study for PP4 rather than PS4. So although the study does achieve PS4 strong, as per the ENIGMA recommendation, we're only using it for PP4, and for that it achieves PP4 moderate. It's worth mentioning here that the study includes data from UK Biobank as well. So if you're using Zanti et al. For PP4, you can't use data from the UK alongside it. So that's the overall piece of evidence from this study is PP4 moderate and not using PS4. And we've seen only 2 participants out of 53 who applied PP4. So 53 out of 207 applied PP4 at any strength for their classification, and only 2 of them have applied the Zanti et al. Many of the participants applied Lee et al., which we will have a look at when we look at the CanVIG application for PS4 and PP4. So it seems that the study is less well known, but hopefully after this, more people will be looking at using it. When it comes to CanVIG, one of the amazing resources that we have in the UK, because we have the NHS and we have the NDRS, which is the National Disease Registry Service, a lot of data from cases, for example, with breast cancer gets submitted to NDRS, which also is presented on CanVAR. And following the CanVIG guidance, we allow the use of the NDRS case data when controlled against UK Biobank in order to perform a case control analysis and to be used for PS4 evidence when available. So again, if you go to the CanVAR UK website, you will be able to access the case frequencies. So you can see here that in the wide proband population, this variant has been seen 6 times. Then if you compare that to the UK Biobank, also European non-Finnish population, which is allowed as a case control analysis in CanVIG guidance, and apply PS4 calculations, I like to use the Turnbull Lab calculator, which is available from the link here, you will achieve a really good odds ratio of 18.3 with a really good confidence interval, and it provides you enough evidence for a very strong PS4. However, we do have a cap on PS4. If you have 6 or less cases observed, then you cap the strength at strong, and hence this variant achieved PS4 strong. And then when it comes to PP4, there is the Lee et al., which many of the participants used within ENIGMA as well to apply PP4 supporting, and that's all correct in that sense if you don't apply Zanti or if you missed Zanti, but the Lee et al. From 2020 does provide enough evidence to apply PP4 supporting. So overall, very few people used PP4 moderate within the ENIGMA VSEP, and very few people used Zanti et al. And then within CanVIG, it's appropriate to use PP4 supporting alongside PS4 strong following the NDRS data. And nearly half of the participants got that correctly. I just now, in the last couple of slides, want to reflect on a couple of evidence criteria that have been applied by some users and why we didn't apply it. So one of them is PM1. We had 48 participants applying PM1. This is not applicable using the ENIGMA guidance, and within the CanVIG guidance, you're unable to apply PM1 because this variant had also functional data applied as part of the classification. So if you look towards the end of the CanVIG guidance, you will see combination tables which states some combinations that we recommend using or not using, or avoiding using, or sometimes capping the strength in order to prevent double counting or reusing the same evidence in different criteria. It's worth looking at it if you haven't. And PM5 is another one. Many users, 67 participants applied PM5 at various strength. That's again for this variant not applicable within the ENIGMA guidance, and within the CanVIG guidance, the combination will be disallowed because the reference variants at that amino acid position all need PS3. Or the ones that we looked at required PS3 functional evidence in order to reach their classification, and so under the combination criteria, this will be a restricted use. And that's all for variant 1. Thank you very much.
Thanks, George. Then I will continue with variant 2. And I hope you can hear me. I guess
yes.
that's
yes, we can. Thank you.
Great. Okay, and this variant is also a variant in BRCA1 and also in actually the same domain as variant 1. But this is a synonymous variant that we are looking at this in this case. And if we look at the summary evidence, this is a variant that is not is absent in NOMAD, and it's also been classified in ClinVar and BRCA exchange as likely benign, and in ClinVar it's classified by the expert panel. And this is a summary of the applicable criteria, and in both using the ENIGMA guidelines and the CanVIG guidelines, the classification ends up as being a class 2 variant, and I will come back to the criteria used. But just to sum up the summary of the participants' classifications, as you can see, most participants classified this variant as a class 2 variant, but there's also quite a few participants that actually classified it as a class 1 or a class 3. And if we look at the guidelines used, most use the ENIGMA guidelines, but also the CanVIG guidelines are used. And if we look at the numbers for the participants, as I stated, most classified this variant as likely benign, but also quite a few as a variant of unknown significance and benign, and even a few as pathogenic. And the criteria used, as this is a variant that are not reported in the NOMAD database, PM2 can be applied, and as you know, the ENIGMA apply this criteria as supporting, while the CanVIG you can get moderate, and in this case the variant is not reported in NOMAD, and therefore CanVIG uses the PM2 moderate criteria, and you can see still people using the ENIGMA guidelines use the PM2 at moderate level. So it's just to remember that a lot of the ClinGen, I guess most ClinGen guidelines use PM2 as supporting. And the other criteria applied is the functional criteria, because this is a variant that has been examined by the Finlay study, the saturation genome editing study, and the variant is also mentioned in table 9, as you can see here in yellow, and it states that you can use BS3 strong in this case. So this is actually the two criteria that can be applied. So you have one benign strong, and you have one pathogenic, either supporting or moderate. And as you also saw in the numbers, some participants did classify this variant as a benign class 1, and I guess most of the ones the participants that have done that has also included the BP4 and BP7 criteria. And if you look in the guidelines, BP4 for BP4 to be applied, you have also to check the variant in the Splice AI algorithm program. And if you check this variant, the score is actually above 0.1, and therefore BP4 cannot be applied. And when you look at the BP7, BP7 can only be used if BP4 is met. So therefore, BP4 is not met, and BP7 cannot be used in this case. And I guess that's one of the problems for participants classifying this variant as a class 1 variant. In contrast, the participant classifying this variant as a VUS, my impression is that it sometimes can be difficult combining criteria in contrast to using points. And that could be one explanation that people actually classify this as a VUS, because you have one benign strong, and then you have the pathogenic supporting or moderate. And if you use the combining criteria, this would end up as classifying this variant as a VUS. But when you have both pathogenic and benign criteria, it is suggested that you use the point system. And using the point system, you will get -2 or -3 points, and therefore the variant should be classified as likely benign. And then I have one last point that people applied and also have applied in the other runs, at least for the variants that I have looked at. That's the BP7, where you use databases to include this BP6 as a criteria. And as you can see here, 41 participants used BP6 at different levels. And it is stated in the ENIGMA guidelines that, or the ClinGen, that this criteria is not applicable, and that also goes for PP5. So that's just to mention that that this has to be remembered. And I guess that was the last slide for me. So over to next variant 3.
Thank you. Next slide, please. So the third variant is also a variant in BRCA1. It's an intronic variant predicted to impact splicing. Next slide, please. So this variant is absent in NOMAD version 2.1 and 3.1 non-cancer, as well as the NOMAD 4.1 non-UK biobank. It is found in 2 males in the NOMAD version 4.1, so that's a bit of a typo. It says females, but it should be males. It's reviewed by ClinVar as being pathogenic, reviewed by the expert panel. It's also in BRCA exchange as being pathogenic, and in silico predictions predict this variant to impact splicing. Next slide, please. So, thank you. Okay, so using the ENIGMA VSEP guidelines, we use the criteria PM2 supporting, PP3 supporting, and PP4 very strong to get a classification of class 5 or pathogenic. Next slide, please. Using the CanVIG guidelines, we get to the same classification, but we use some additional evidence. So using CanVIG, we can use PS4 at strength of very strong, PM2 at supporting, PP3 at supporting, and PP4 at very strong. Next slide, please. So in general, people did very well with this classification. 248 out of 287 participants classified this as class 5, whereas 31 classified this as class 4 likely pathogenic, and 7 classified this as a VUS, and 1 individual classified this as benign. Once again, ENIGMA was the classification scheme most individuals used, but CanVIG was also used by quite a few. Next slide, please. So a number of individuals used either PVS1 RNA or PS3 because of some splicing studies that are published on this variant. So when we look at the ClinVar guidelines, they recommend for splicing assays to not use PS3 or BS3, but rather to use PVS1 RNA or BP7 codes. And then they give a table of the expected results. So depending on the assay used, what threshold is needed for the study to be significant. So for example, if it's a patient with a quantificational study, you want less than 10% of the wild type allele left and more than 90% of the transcript being the non-functional transcript. If you're using the mini gene, and you can use that at full strength, so PVS1 RNA at whatever strength is appropriate. You're using the mini gene assay only, you have the same thresholds of less than 10% wild type and more than 90% aberrant transcript splicing. However, the strength is decreased by one level, so PVS1 strong would be the max. Additionally, looking at patients with quantification, if you have a range between 10 and 20% wild type residual expression, and between 90 and 80% aberrant transcript functions, you also downgrade this by one level of strength. And then mini gene assay alone in this threshold of 10 to 20% wild type transcript remaining, you would downgrade it by 2. So instead of PVS1, you'd downgrade it to PVS1 moderate. Next slide, please. So when we look at the data available for this variant, there are 3 functional studies that look at splicing. The first study by Theory et al. This is a mini-gene assay. And what they showed with this mini-gene assay is that almost all, or if not all, of the transcript available is aberrant. It includes an extra 10 nucleotides. The second assay is by Wiley et al. This is a RT-PCR from a lymphoblastoid cell line created from a patient with this. And they needed to perform sequencing to actually see the change, but they also see the same 10 base pair increase in the majority of the transcript available. And then there's a third assay published in 2019. And this assay is sequencing of cDNA from patients. And what they find is they also see some aberrant splicing, but they see the majority, so over 70% of the transcript being expressed is the wild type transcript, with only 30% being aberrant. So there is a contradiction here. All 3 assays are showing aberrant splicing because of this variant, but one of the 3 assays isn't meeting the threshold of being over 80% or over 90% abnormal transcript to qualify for PVS1 RNA. And based on this contradiction, this doesn't qualify for use of PVS1 RNA at any strength. Next slide, please. So the second point is the use of PS4 case control studies. So 37 out of the 287 participants use PS4. As my colleague went over earlier, case counting is not applicable using the ClinGen guidelines. However, using CanVIG-UK, you can use the National Disease Registry Services and the UK Biobank data to perform a case control study. And so I also like to use the Turnbull Lab PS4 calculator. And so you can find this data in the CanVIG-UK database. So if you look up this variant in CanVIG-UK, you can pull up all the data from the National Disease Registry as well as the UK Biobank. And in this particular case, we have 15 out of 66,000 white cases and zero females from the UK Biobank control data set from the non-Finnish Europeans. And if you plug this into a PS4 calculator, you get to a strength of PS4 very strong. Next slide, please. This was also touched upon, but the use of PM2 differs slightly between the CanVIG-UK guidelines and the ClinGen guidelines. So in ClinGen, PM2 is always used at supporting for the BRCA 1 and 2 variants, if applicable, whereas with the CanVIG-UK guidelines, it can be applied at moderate if it's completely absent from NOMAD version 4.1. However, if it is present in at low frequency in NOMAD version 4.1, it can be applied at a level of supporting if the frequency is less than 0.002%. So in this case, this is where it's applicable because there are 2 individuals in NOMAD version 4.1 with this variant. Next slide, please. My last point is the use of multifactorial likelihood clinical data. So this variant is found in 2 different studies, Lee et al. And Easton et al. When we combine the 2 likelihood ratios, we get a combined likelihood ratio of 117,612. When we convert this to the log likelihood ratio, we get a significance of very strong. So you can use PP4 very strong. Additionally, the ClinGen entry in ClinVar has internal data which they've included in their likelihood ratio, and they get to a score of 10,799,000. So this is even well above what you get by combining these 2 studies. However, this is all internal data, and that's how you can use PP4 at a level of very strong. And the majority of participants who classified this variant did use PP4 at some level of strength. So 245 out of the 287 participants did use PP4. And I'd say 2/3 of those, 178, used it at the appropriate very strong, whereas about 56 used it at strong, 4 at moderate, and 7 at supporting. Next slide, please. And I'll hand it over to my colleague.
Thank you, Andrew. So for variant 4, can I have the next slide, please? Yes. So it's again a variant in BRCA2 at nucleotide position 9242, where a T is replaced by a C, and this leads to an amino acid change at position 3081, which is located in a functional domain. It's within the DNA binding domain. Next slide, please. So there are some data about this variant. There is the GNOMAD. It is present in all versions of GNOMAD, so both in version 4.2 non-cancer and version 3 non-cancer. Also, it has been reported in the UK Biobank, also in female carriers. In ClinVar, the data, the classification data are conflicting. So there is 3 labs reporting it as uncertain significance, 1 as benign, and another 9 as likely benign. In BRCA Exchange, it was not yet reviewed, and there are also the in silico predictions. Many are listed here, and you can see they vary from benign, likely benign, to possibly damaging or probably damaging. Next slide, please. We have classified this variant as a class 1, using both the VSEP ENIGMA guidelines as well as the CanVIG guidelines. However, as you can see, with the CanVIG guidelines, we have applied, there was one criterion, the BP4, that we could not apply. And I will explain in the next slides why. Next slide. So the classification of this variant went globally very well. So almost 50% of the participants classified this variant as benign, about 36% as likely benign, and a smaller proportion as a class 3, uncertain significance. However, it's notable that none of the 287 participants classified the variant as actionable, and that's actually what we want, that pathogenic, well, likely benign variants are not misclassified as actionable variants. Next slide, please. So the first criterion we have applied was about the functional studies, and this variant has been extensively tested in several studies. You can find the guidelines when to apply the functional studies at what strength within the VSEP guidelines. And there is this nice table 9 that was also shown by Thomas, if I remember well, where they have an Excel table listing a large number of variants. They also provide the tests with which they were tested, and they provide you with the assigned code and the code weight. According to CanVIG, they also have this gene-specific guidelines, and within the gene-specific guidelines, it's nicely listed which functional assays you can use at which strength. And on top of that, they have the very nice CanVIG-UK database, where there is a nice summary of the functional of the results of these functional assays, so that you don't have to dig into all supplementary tables of these functional assays yourself. And you see that, according to both guidelines, you can apply BS3 at a strong level. For VSEP, it's based on the Richardson paper that was published in 2021. And for the CanVIG guidelines, they refer to the Hu et al. Paper from 2024, where it's occurred as it behaved like a normal variant, and in the Hart et al. Paper, it behaved as a neutral variant. So that's very, very good. And can be applied at strong. Next slide, please. The other criteria we have applied was BP5 strong. So I excuse myself for the little typo that's in the title of this slide. So it should be BP5 strong and not BS5 strong. According to the VSEP guidelines, we can use this criterion based on likelihood ratios that are calculated from clinical data, and VSEP has provided some cutoffs for this. How can you calculate yourself these likelihood ratios? Well, if the variant is listed in BRCA exchange, there is a nice table from the Parsons et al. Study where, and that was a mega multifactorial likelihood analysis, and you can calculate yourself the combined likelihood ratio by amplifying the likelihood ratio for co-occurrence, segregation, family history, pathology, and case control, and then you receive a value of 0.0139, and this is below the 0.05, and then you can classify it as strong. In CanVIG, this can also be applied as BP5 strong. For the likelihood ratios, it looks a bit different, but in fact, these are the same cutoffs as for VSEP. And also here, the CanVIG-UK database, it's very easy to look up the values. They have already calculated for you the combined likelihood ratios, and they've also calculated the log likelihood ratio, so you can consider it as points, and this is so minus 5.84 points. So this is also at the strong level. Next slide, please. Then another criterion we have applied is the BP4 supporting, and it's based on the in silico predictions. So according to VSEP, you have to use the Bayes-Dell prediction tool, and you can apply it when the variant is located inside a clinically important functional domain. And this is the case for this variant because it's within the DNA binding domain. And there should be no predicted impact via protein or splicing. So it should be splice AI should be lower, should be lower or equal 0.1. For this variant, the Bayes-Dell score reaches is lower than the 0.18. CanVIG, however, applies another prediction tool. It's the Revel that they suggest to use. And to be able to apply BP4, you should reach a value that is lower than 0.4. However, the Revel score is higher. It's 0.627. Therefore, it is not applicable. And you can also not apply PP3 because then the cutoff is 0.7, and this is not reached either. Next slide, please. So there are some criteria that have been applied by the participants and that we as a panel did not apply. The first one is the PM1. As you can see, it was applied by the participants at variable strengths. In the VSEP, it cannot be applied at all because it's considered as a component of the bioinformatics analysis, and we have used the BP4 criterion. And then in CanVIG-UK, PM1 cannot be used where functional data are being used, and this is the case. We have used the BS3 functional data, so PM1 cannot be used. The second criteria is PM2, and this is about the prevalence and the frequency in databases. For VSEP, there has been published in 2020 already that it should be maximally used at supporting levels, and there was still participants using it at higher strengths. According to the VSEP guidelines, the variants should be absent from controls in an outbred population, and the guidelines are still focused on GNOMAD version 2 and GNOMAD version 3 non-cancer. In CanVIG-UK, they have they applied the GNOMAD 4, the latest version of the GNOMAD database, and the variant is present in all the versions of the variants. But when I was preparing these slides, I realized that CanVIG-UK has a general cutoff of 1 in 50,000. So in theory, you could also, I think for CanVIG, you could actually still use the PM2, but not at the moderate level, but it should be at a supporting level because the variant is is present in the UK biobank and in the non-UK biobank. Next slide, please. Then PP3 was used by a proportion of the participants, and PP3 is a prediction tool. I checked because the majority used the benign tools, but some of the participants explained that they used the meta or an N tool where it is within the range of supporting pathogenic. However, this is not the tool that should be used. According to VSEP, you should use BIAS-DEL, and according to CanVIG, you should use Revel, and it does not reach for Revel, it does not reach the cutoffs, and for BIAS-DEL, you reach the cutoff towards benign and not pathogenic. Another criterion that was applied by 27 participants at different strengths was the PP6. So it refers to reputable sources like ClinVar, where it's reported as benign. However, both VSEP and CanVIG-UK advise not to use it. It's not a valid code anymore. And if you want to use it, you should look at evidence and then use this evidence under the right parameter. Next slide, please. BS1 strong was applied by about 26 participants, and this is about the allele frequency that is greater than expected for the disorder. You can use it both VSEP and CanVIG. However, you should reach a certain frequency, and this was not obtained for this variant. So you see here the cutoffs, the filter allele frequencies for the strong and the supporting level in VSEP. However, this is not reached for this variant, especially not in the non-cancer subcohorts of version 2 and 3 of GNOMAD. For CanVIG-UK, BS1 can be used as strong when the group max filter allele frequency in GNOMAD 4.1 is higher than the maximum tolerated allele frequency for BS1. However, this is not obtained. So in GNOMAD 4.1, the filter allele frequency is 0.508. 030, and this is lower than the cutoff that you have for BS1. So that's why you can definitely not apply this one. Next slide, please. Can I have the next slide? Yes, thank you. There was another, then there was a few criteria applied by a few participants. So the BS4 strong, it's about segregation. And in VSEP, you can use it when the likelihood ratio is lower than 0.05 at strong, and also at very strong if this likelihood ratio is even lower. You can look up the segregation likelihood ratio in BRCA exchange, but it does not comply with these cutoffs. And in CanVIG-UK, you cannot use it because it's incorporated in the BP5 score. Next slide, please. Then there were a few participants who used BP1 strong. So this is about missense variants in genes where primarily truncating variants are known to cause disease. This was the original formulation of this parameter. However, this is not, this cannot be applied. This is not interpreted anymore like this in the current guidelines. So you can only use BP1 strong for variants when they are located outside of important clinical functional domains. However, this specific variant is located in the BRCA2 DNA binding domain and cannot be applied. Next slide, please. And then some participants combined it with BP7. However, according to VSEP, this variant is for intronic and silent variants, and can be for missense variants when they are located outside a clinically important domain, but this is not the case for this variant. I pasted here the figure 1A from the VSEP guidelines, where you have a nice diagram that you can follow for missense variants where there is a predicted impact on splicing, which is not the case for our variant because the splice AI is lower than 0.1. It is not. It is in the functional domain, and there is a predicted impact on the protein, and this is not the case. And then we apply BP4 as we have discussed before. For the BP7, it cannot be applied at the supporting level for this variant because BP4 is met, and then you do not apply BP7. And for CanVIG-UK, this variant is only used, this parameter can only be used at the supporting level for synonymous and silent variants, for which there is no impact predicted by prediction programs. Next slide, please. And then BP2 was applied by a few participants, and this criterion has to do with pathogenic variants in dominant genes. And when you have B allelic inactivation, it can give another phenotype. In VSEP, you cannot apply it because it's not applicable. When there is no Fanconi anemia phenotype. And for CanVIG-UK, you can use it if it's in trans with a pathogenic variant. And then some of the participants really argued why they have used it, and they wrote down that it was reported to co-occur with a pathogenic variant. They listed this variant, and this was in the UMD database. However, when I read this, for me, it's actually insufficient information to judge if these variants are really in cis or in trans, and then you cannot apply it. I checked which criteria the applicants applied, and there may be older guidelines where there was not such, where these details were not listed. And I think we are now at this was the last slide, and I hand over the words to the next speaker.
Thank you, Kathleen. Next slide, please. So the fifth variant is a frameshift variant in BRCA2. Next slide, please. So this variant is absent from NOMAD 2.1 and 3.1 non-cancer cohorts, as well as being absent from NOMAD version 4.1. In ClinVar, it has a 2-star entry, being classified as pathogenic or likely pathogenic, and it is not yet reviewed by BRC Exchange. Next slide, please. Next slide, please. Thank you. So using the ClinGen VSEP guidelines, we can apply the evidence PVS1 at very strong and PM5 PTC at evidence of strong. Using the CanVIG-UK guidelines, we can apply PVS1 at very strong, PM5 PTC at strong, and we can also apply PM2 at a moderate strength. Both of these give a classification of class 5 or pathogenic. Next slide, please. So most participants classified this variant correctly. Overall, we had 240 out of 287 participants classify this as pathogenic, as well as 44 classifying this as likely pathogenic, and only 3 classifying this as a VUS. Next slide, please. So what tripped up some individuals is the use of PM5 for a frameshift variant. So using the BRCA 1 and 2 guidelines, PM5 the code has been repurposed. It no longer applies to novel missense changes at amino acids where a different missense change exists that's pathogenic, but it applies to protein truncating variants in an exon where a pathogenic protein truncating variant exists. And you can use it at varying strengths from strong down to supporting. Next slide, please. So to find what level of strength you would apply this PM5 at, there's a supplemental table 4. It's a standalone Excel file in the BRCA 1 and 2 guidelines. And you open it up and then you find your exon of interest. And this table will tell you exactly what strength to apply your PM5 at. And so for this particular variant, PM5 should be applied at a level of strong. Majority of people did use this, but for the individuals that got a VUS or likely pathogenic instead of pathogenic, this was one of the criteria that they often missed. So all 3 individuals who classified this as a VUS did not apply this, and 42 out of the 44 that classified this as a likely pathogenic variant did not apply this. So this might be one of the criteria that would bump you from likely pathogenic to pathogenic. Next slide, please. The other piece of evidence is the use of PM2 absent from control populations. So 181 submissions out of the 287 applied PM2 at some strength. 120 of these were using the ENIGMA guidelines, and 42 were using CanVIG-UK guidelines. So as mentioned before, there are some differences between the ENIGMA and CanVIG-UK guidelines, and PM2 is definitely one of the points where they do differ. Next slide, please. So using the ENIGMA guidelines, PM2 supporting is not appropriate for insertions, deletions, or indel variants. Whereas, so therefore for this variant, PM2 should not be applied at all. Whereas using the CanVIG-UK guidelines, they do not have such a restriction. And so the reason why PM2 supporting is not appropriate for frameshift variants using the BRCA 1 and 2 ENIGMA guidelines is because there's questions about the quality of the data in NOMAD. Next slide, please. So they referenced this publication where they looked at the precision and recall of NOMAD data for both missense variants and for frameshift variants. And you can see for precision and recall, it's well over 90% for as long as you get over 20x coverage. Whereas when you look at the recall for frameshift variants in the NOMAD data, it drops down to 40%. So there is a concern because of this low recall that it's possible the reliability of the frameshift data in NOMAD is not at the level that we would want to be using it to apply PM2. And that's why PM2 supporting is not appropriate for frameshift variants, according to the ENIGMA guidelines. Next slide, please. And now I'll hand it back to my colleague, Beth.
Thanks, Andrew. So this is the last variant that we're going to classify today. Do I have control of the slides? Perfect, thank you. So this is a copy number variant in the PALB2 gene, corresponding to an exon 7 to 8 deletion. See some different ways of writing that with different nomenclatures there. These are some findings in various databases. I'll hopefully have time to just talk through them in a moment, but I'm trying to whiz through so we have some time for questions. So the expected classification for this is pathogenic, and that is regardless of the guidelines that are being used. The CanVIG guidelines actually recommend just deferring to the VSEP guidelines and just offers a few clarifications. So the expected codes and strengths that we were hoping participants would apply are PVS1 at very strong, PM2 at supporting, and this PM5 PTC adjustment at a supporting strength. Most participants did really well on this, with 96% of participants classifying the variant as actionable, as pathogenic or likely pathogenic, so getting an appropriate outcome for the patient there, with 54% reaching the expected classification of pathogenic and 43% reaching likely pathogenic. And the main difference between those two outcomes seems to be in the application of PM5, which we'll talk about in a moment. So I will try not to dwell too much and maybe expand on this a little bit in the summary document if people are interested in the nitty-gritty of looking at copy number variants, but the majority of participants applied PVS1 correctly at the very strong level. So I believe that most people will probably have a very good understanding of this. But in order to do this, we considered whether the loss of these exons is predicted to result in a frame shift or a preservation of the reading frame. There's a number of different ways you can do this, depending on the resources available to you and how your brain works. You can rely on this lovely figure from the VSEP guidelines, which allows you to visually see whether removal of certain exons or inclusion of certain exons is predicted to result in frame shift or frame preservation. Our exons here, you can see, have designated anti-parallel lines, which signify a frame shift would result in the loss of those. You can also sort of investigate this from first principles if you have access to a tool that enables you to visually inspect the start and the end of your exons of interest. So you can see here the same conclusion would be reached looking at those overhangs at the end of your region of interest. If you don't have VSEP guidelines like we do here, or maybe tools available to you that allow you to visualize the exons, you can also do this in a sort of mathematical way, looking at maybe resources like Canva, where you can get the exon length of the region that you're interested in and determining if a frame shift is predicted based on whether that's a factor of 3 or not. So no matter which way we investigated this, we can then follow our VSEP decision tree laid out there, showing we have a deletion that is predicted to result in the disruption of the reading frame, and we can then apply PVS1 at that full strength, which many people did. So next we investigated PM2. Now we were expecting people to apply this at supporting level, and that's limited as per the VSEP guidance recommendation. So can't reach that moderate strength for this code. And this is on the basis of the frequency in NOMAD version 4 dataset being under this 1 in 300,000 threshold. And because this is a copy number variant, we need to look into these either SVs and or the CNVs version of NOMAD, where these types of variants are recorded. You can have a look for some information in the info bubble about the types of variants in there, and this snippet here in grey is taken from CanVIG, which explains the sizes of your variants and which databases they will be found in. This PALB2 exon 7 to 8 deletion is a variant smaller than 3 exons, so we need to investigate in the SV database only, and this screenshot is taken from there, with the little box highlighting our region of interest. And we can see visually, but also there's a table you can inspect that this variant is absent, as are similar or overlapping variants in that region. And so we can apply that PM2 supporting. Just another note, if you're unfamiliar with working with CNV, is just to be careful when you're using this database. If you're visually inspecting this graph, check your direction of the gene and your exon positioning and your transcripts to make sure you are in fact looking for the correct region that you're interested in. Look for similar and overlapping variants, and be wary of you using the correct genome build if you're referencing other programs that you may be used to using different builds. And it's 38 for NOMAD 4. So then the last piece of evidence we were hoping people would apply is this PM5 PTC for premature truncating or protein truncating codons at the supporting level. And this, as I said earlier, was the main difference between participants who reached class 4 and the expected class 5 outcome. So in green, this is the information here from the VSEP about the application of this code. So it is applicable to frame shifting or truncating variants with premature termination codons upstream of that tyrosine 1183. And although our variant is an exon 7 to 8 deletion, and we have an anticipated frame shift that would result from that, we understand that it could be interpreted a little bit with some ambiguity, because the guidelines haven't been explicit about using this for copy number variants. So we were happy with the sort of assumption that there was going to be a frame shift and a premature termination, and this code was applicable, but just had some clarification from the opinion of someone on the PALB2 VSEP group, which confirmed this, that they were happy that if PVS1 was achieved at that very strong weight, and this would still be applicable for copy number variants in this way. So nice to have clarification. We didn't apply any other codes, but just one quick slide talking about whether you would gather any other evidence and where you might find that. So a reminder that proband counting isn't permitted by the VSEP for PALB2. However, you may have still searched various databases to look for other types of evidence and for opinions on your variant. So this can be quite challenging for copy number variants. So just to give a couple of examples of how you might do this, searching on ClinVar can often be difficult to bring back records matching your variant of interest, but it can be helpful to use this visualization tool, again, where you can filter on size and type of variant and the position of your variant to try and narrow down if your variant's been reported previously. It can give you some nice references and some nice evidence. In this case, we found 2 records matching our variant of interest, both classified as pathogenic, which is reassuring. This is a snapshot from LOVD. Again, can be quite difficult to know how to search for your copy number variant. It can be useful to search using the first and the last nucleotide of the exons that are affected by your copy number variant. That can often bring back different records that may match your variant. And as you can see, there's one record here with some specified breakpoints, whether that's been clarified or sort of set by this submitter's assay, but it would encompass the region of interest of our variant. So even though we may have not searched specifically using this nomenclature that's provided, you can still find useful references. And this is just from HGMD as well, again showing the different way that different copy number variants may be recorded, and you want to maybe take a little bit extra time to carry out your literature search if you're gathering evidence, to make sure that you haven't missed anything useful that you could use. And that's the end of that. I'll pass back to, I'm not sure who I'm passing back to.
Sandy.
Yep, it's me. Thank you very much. Hopefully you can hear me. So thank you, all our experts. We've got lots and lots of questions coming in, which is absolutely brilliant. Thank you very much. We're going to put them on the spot very soon, so keep them coming in. It's always nice to have an overview of the submissions and the different classifications. And we do, oh, can we just go back to the last slide, please? So we do like our bubble plots. So the larger the bubble, it means that the more participants classify that variant. The classifications are up on the y-axis, and the x-axis is our variant 1 to 6. So the expected classifications are in our lovely raspberry ready colour here. And you can see that the other classifications that were submitted is in the golden colour. And I think the take-home message here is some of the other classifications, the bubbles are very small, which is indicating that we're getting much better consensus and agreement across the board. Variants 2 and 4 obviously were the more challenging ones, and we've had great amount of in-depth detail as to the background why. And there's quite a few questions coming in as well around particularly variant 2. So let's move on to our live Q&A. So next slide, please. Thank you, obviously, to all of our experts that's been on this panel today, and also to our guest speakers from the first webinar that was before Run 16 opened. So next slide, please. Summary report for this run that we've discussed will be available later this month or early July. We're just collating the information, and obviously we will answer all of the questions that were raised either in the webinar 1 or this webinar today that have been submitted by participants, so everyone can see what has been discussed and the expert point of view from those. Next slide, please. Thank you very much for your attention for the webinar. We'll now move on to the Q&A. Next slide, please. Right, so Simon, do you want to kick us off?
Yeah, will do indeed, Sandy. Thank you very much indeed to that. And thanks once again to our 5 speakers today. Great job, guys. Thank you. Right, this is always the most difficult part of the whole process for our speakers, because obviously your questions are coming in live, and much of it's very technical in nature. So please do bear with us as we try to work through your list of questions. I will say that if we don't get around to answering your questions today, we will ensure that we've got a copy of the questions and answer them via the summary report anyway. So much of what you'll hear today will also be included in the summary report. I think looking at the questions, Sandy, there's been quite a lot of questions. I think we'll probably start with variant 2 for Thomas, because there have been a lot of questions coming in, all of which effectively say, why have you classified it as a likely benign variant versus a VUS? So I think I think in general they're all focused on that. So Thomas, are you able to give us an overview of question 2, please? Variant 2.
Yes, and it's a very relevant question, because actually we have had some discussion going back to our notes from our meetings, and we actually agree with the questionnaires that using the CanVIG guidelines, it is actually stated in the general guidelines by Garrett that you have to have 2 concordant evidence criteria to use or to classify a variant as likely benign or likely pathogenic. So actually using the CanVIG guidelines in this case would classify the variant as a VUS, a class 3 variant. So in the questionnaires are actually right in this case. So using the CanVIG, it is a VUS. So we have to give credit to the people making these questions. That's for sure.
I think also emphasises just how difficult this is, isn't it, Thomas? Because even within our expert group, we can't always get it completely 100% right.
Yeah, and actually it seems like in a lot of classification schemes, you are going to the point system only, and this is a question that will pop up for sure using the point system.
Yeah, totally agree.
And what we can do, Simon, just to come in there, we will look at the system, the platform, and look at how we can address and accept anyone that's put a VUS in as an answer as correct as well. So we can adapt those. Thank you.
Yeah, thank you very much indeed. Thank you, Thomas. Sandy, I'm going to hand over to you for the next question.
Yeah, great. Thank you. I would like to ask Andrew around case 3, his variant there. So one of the questions is, why can't you use PS4 for ENIGMA? This is on the recent ClinGen guidance. And the ENIGMA guidance talks about using case control data for PS4 that's ethnicity and country matched to the control data set. And I think it's relevant to variant 1 as well that George talked about.
So I believe this question's in reference to being able to use PS4 with following the CanVIG guidelines. We did have some discussions with our CanVIG colleagues because this had come up when we evaluated the variant. Using the ClinGen guidelines, we felt the case control study needed to be a published case control study and not a match of control data set and a case data set. But I do understand why individuals might feel this is appropriate. I don't necessarily think it's wrong. We just, we took a more conservative view of the ClinGen guidelines, and that's why we didn't use PS4 by using the UK Biobank registry and the disease registry. So there was some debate amongst us whether it was appropriate, but we thought to err on the side of caution.
Great, thank you, Andrew. I see George wants to come in. George, what's your thoughts?
Yeah, so if you just read the ClinGen guidance on the face of it when it comes to the calculations, it does just state ethnicity and country matched, which with UK Biobank and choosing only the white European NFE population, it may sound like it, but we know from various discussions between the CanVIG group and ClinGen that they don't prefer using it. It's not a specific matched case control study, but also they had concerns about the assertment of the NDRS data because the breast cancer cases are not unselected cases. They have been selected after matching enough criteria to be eligible for testing in the UK. So they are selected breast cancer cases, and that did not match with the way the case control analysis is considered by ENIGMA. But that's a very technical detail, and reading the guidance on the face of them in terms of the ClinGen guidance, you may get the impression that it's appropriate to use it against the UK Biobank ethnicity matched data.
Thank you, both of you, for covering that. Simon, there's a couple of questions come in for variant 6, our CNV for Bethan. Will we talk about those? I'm aware of time, but I think it's quite interesting because that's the only variant. There was a copy number variant.
Absolutely.
So for the PALB2 exon 7 and 8 deletion variant, the breakpoints are not known, which means that an unexpected effect on RNA cannot be excluded. So therefore, they kept some uncertainty in the classification by not applying PM5 supporting and keep it as a class 4 instead of a class 5. Bethan, do you want to have some comments on that, please?
Thank you. Yes, I was just trying to reply to that one quickly. Yeah, I think it's a great point, and that is why a lot of the VSEPs, most of the VSEPs don't allow application of PM5 for splice variants, even when they're predicted to be completely loss of function, because of that uncertainty around that impact on splicing. I think in this case, with a CNV that's predicted to be disruptive, you could well apply cautiously if you weren't sure, and I think that's some of the ambiguity and the uncertainty that we were expecting to come up, which is why we also reached out to that to the VSEP group to sort of try to get that clarification, and just to give ourselves a little bit of certainty as well about how that might be applied. But I think that is a good point, and the more cautious you are, I tend to try and be cautious in my application as well. You don't want to overestimate the strength of things, so I think it's really good that that's considered.
Thanks, Bethan. And I think that's probably a take-home message, isn't it, for other CNVs right across the board about that uncertainty when you don't know exact breakpoints. Great, thank you. Simon, back to you then, please. Thank you.
Thank you, Sandy. I'm conscious of time, so I think we've got time for one more question, which I'm actually going to address to you, Sandy, specifically. And I think this is really more, if you like, an operational question, but I think it also addresses the point we made about case 2, about being able to give an alternative description of a variant. So the question is saying, when submitting classifications, would it be possible for a free text box to be visible next to all the evidence codes, not just the ones where an evidence strength has been applied? For some variants, evidence codes may be met but are not appropriate to apply. And would it be possible to allow for further evidence of competency to be documented in the submission report? Sandy.
Yeah, I mean, I think this is the system evolving, isn't it? And it's great to get this feedback. So yes, we'll definitely look into that. I think it's about building your record of your own evidence of competency, which is what the whole platform's around, the Genie platform. So we'll look into that. And obviously it will get updated as we see the ACMG version 4 guidance coming along as well. So yes, watch this space for new developments. Thank you.
Great. Thank you. I'm very conscious of the time. We are now overrunning, but so I think I'm going to draw it to a conclusion for today's webinar. A big thank you to you, the participants, for dialing in. We've had a large number, over 150 dialing in today, which is great to see. As Sandy said, we will be updating the summary scheme report with the information we provided. And where we haven't got around to answering a question today, we'll obviously try to include that in the report as well. So do keep an eye out for that. Webinar will be available hopefully in a couple of weeks' time. We will let you know via the various media channels from both EMQ and GenQA. And finally, that leaves me just to sort of thank our 5 speakers today. Thank you guys for such a great job. It's not easy, as you can see from today's feedback. And we really do appreciate the time and effort you put into answering the questions and providing the feedback to the participants. Thank you also to the AZ, MSD, Indigo medical teams for all their work in supporting this. And finally, thanks to my co-chair, Sandy Deans. Great job. And we'll look forward to catching up with you at another webinar sometime soon. Thank you very much indeed, and have a good rest of the day.
Thank you, Simon. And can I just say a quick thank you to Angie as well, who actually manages the Genie platform and has done a fantastic job working with our expert advisors. So thank you, everyone. Bye-bye.
Thank you. Bye for now.